Okay, thanks to Stephen and Chris for inviting us to come along and talk about how we apply biophysics in drug discovery at the end of your lecture series on biophysical methods. So hopefully this will be a little bit of an overview on how we use some of the methods that you've talked about already in the drug discovery process. So what we're going to cover today, I'm going to do an introduction to the drug discovery process and show you how and where some of the biophysical methods fit into that process. And then Chris is going to take us through the application of those biophysical methods in the drug discovery process and in several different areas. So in reagent assay quality control, in how we use biophysics for primary screening to find initial hits, and then to cover how we might use biophysical methods in orthogonal screening, so to give us confidence in those initial hits that we found from other potentially biochemical screening methods. And then a little bit around how we use biophysics to do mechanistic characterization, so trying to understand how the compounds that we found are binding to the target protein, what are their kinetics and thermodynamics, whether they need other compounds, other ligands in order to bind with the highest affinity. So I'll start first with the drug discovery process. Okay, so this is a simplified overview of the drug discovery process. We start off with a disease for which there is an unmet medical need, and then we try to identify a target macromolecule whose function we can intervene with in terms of trying to modify the disease process. Then we start off by looking to identify some potential compounds, usually through some sort of primary screening approach. And then we go through several rounds of chemical optimization through the hit to lead and lead optimization process to improve the potency, pharmacokinetic properties, and physicochemical properties of those compounds, until we get compounds that are suitable for going into preclinical testing in animal models, and then hopefully into humans, phase 1 to phase 3 clinical trials. And the ultimate aim at the end is obviously to end up with a marketed drug. So this is an overview of the drug program operating model, and you can see it takes a long time to go from initial target identification to having a product launched at the end. It takes, on average, around 10 years, costs between $1 and $5 billion to take a project all the way from initiation to marketed drugs. And many projects, most projects, actually fail. So around 1 in 100 drug research projects actually leads to a marketed drug at the end. And what I've shown at the bottom is where biophysics contributions are most commonly applied. So usually in the early phases of drug discovery, so the phases that are preclinical in nature. And I'll give a bit more detail on that on this slide. So usually biophysics methods are applied following target selection in hit identification and through the lead identification, lead optimization processes. And the sorts of things that we use them for are shown here. So often we want to understand our protein. So we'll do some protein QC with biophysics methods. And alongside understanding the protein, we also want to understand how potential tool ligands might behave. So these might be compounds that have been identified in the literature, and we might want to use those in order to set up primary screening assays. And so we need to verify that those compounds do actually behave the way they were described in the literature. We can also use biophysical methods to give us an understanding of the assays that we use. So are the assays that we're potentially going to run in terms of hit identification actually measuring parameters that are useful? So we can look at KD values for test compound binding and show that in the assay it has a similar KI value, for instance. And that helps us to understand that the assay is actually performing the way we hope it will. Then we can look at target ligandability. So by looking at a range of different compounds and their affinities and whether they bind to the target protein, we can assess how difficult it might be to actually find hits and leads for those targets. Then we can also use biophysical methods in primary screening itself. So we typically use biophysical methods in fragment screening, where you need a sensitive method in terms of understanding and measuring affinity between a test compound and the target protein. And we can also use similar methods in terms of hit evaluation. So after we've run a biochemical primary screen, we want to make sure that those compounds are actually interacting with the target rather than just interfering with the detection system. And so we can use sensitive biophysics methods to measure that target engagement. And then later on in the process, we want to understand in more detail how our compounds are binding to the target. So this is where we then employ more detailed mechanistic characterization. So trying to understand to which form of the protein the compound is binding, and using full kinetic and thermodynamic profiling alongside structural information, we can get a really nice picture of how those compounds interact with the target protein. And that helps us to make new design hypotheses and drive the optimization process. So just a comment on why it's so difficult to make drugs. So estimates of the number of possible drug-like molecules are around 10 to the 60. And if you think about that in contrast with the number of seconds since the Big Bang, which is only 10 to the 17, then chemical space is actually huge. And trying to find the right compound for your target in that chemical space is actually really difficult. So to give you an appreciation of how long that might take, if we had 10,000 chemists, each preparing one compound every second, it would take 10 to 48 years to make all of those 10 to 60 possible drug-like compounds. So really quite difficult and time-consuming, which is why drug discovery is often likened to finding a needle in a haystack. So, but we're endeavouring to try and do that. So it would be useful to know what a quality drug looks like when we've actually gone through that process. So a bit of audience participation now. What I'd like you to do is just put your hands up if you think that's a quality drug. No, nobody thinks that's a quality drug. Okay, what about that one? Could that one be? Oh, Stephen thinks that one's a quality drug. Good, yeah. What about this one? Anyone like that one? Or that one? You like that one? Okay, that's good. That one? Yeah, okay, excellent. Or this combination of two molecules. Nobody likes that. Okay, well, let me tell you that they're all AstraZeneca drugs, all blockbuster drugs selling more than $1 billion. These numbers from 2021. So as you can see, it's difficult to spot a quality drug. They all look different, different shapes and sizes, and different physicochemical properties. So it's important to be able to recognise once you've got it what a quality drug looks like. So how do we what do we use to try and help us identify these sorts of molecules? Well, this is going back now to some work that Chris Lipinski did nearly 25 years ago now, looking at what oral bioavailable drugs look like. And he came up with this rule of 5. And it's not really a rule, it's more like a set of guidelines which help us to focus on particular chemical properties. And so his guidelines were molecular weight of less than 500. This is because larger molecules tend to diffuse more slowly. A C log P, so this is the partition coefficient between water and oxynol. And a small log P means that the drug is soluble in both oxynol and water. And you need that in terms of drug discovery, because you need a compound to be able to dissolve in the blood and then actually come out of the blood and go through into membranes and tissues. So you need a log P that allows both those things to happen. The other rules were less than 5 hydrogen bond donors, less than 10 hydrogen bond acceptors, and less than 5 rotatable bonds. And so these rules are tended to be broken these days. We're getting lots of different modalities. So I'll give you an example of PROTACs, which have a warhead for a target of interest and a warhead for an E3 ligase, and a linker linking those two warheads together. And so they tend to be larger molecules with less optimal physicochemical properties. But I guess it's still a generally true phenomenon that if you want to have properties of solubility, absorption, and good metabolic stability, that working in this area of Lipinski space is still useful. Okay, so how do we then use biophysics in drug discovery to help us move from an in vitro binding event to something that might be useful in humans? Well, we can use biophysical methods to assess some of these parameters. So affinity, binding kinetics, off-rate, on-rates, binding mechanism, and even potentially enthalpy. And these parameters are associated with things like potency, with structure-activity relationships, with selectivity, can be associated with off-target side effects, and obviously binding mechanism is related to molecular interactions on your target, but also other off-target proteins, and potentially drives the cellular activity that you want to see. So we can see that biophysical methods help us in understanding how to move from in vitro situation to hopefully a drug in the end in a human. But in the early drug discovery process, we tend to ask some of these questions. So is my target suitable for use in the assays that I want to put it in and use to find potential hit small molecules? How druggable is the target? So this is relating to, are there specific pockets on the protein that we can fill with small molecules that might have the desired biological effect? Can I find ligands for that target? So, am I able to use that target in a screening approach to find new molecules that might bind to that target? We can answer the question about a single ligand. Does this tool compound bind to the target? And if so, how tightly does it bind? And how does it bind to the target protein? So does the ligand require the presence of other molecules to help it bind, so cofactors or other ligands that bind to the target protein that form the binding site to which the test compound binds preferentially. And then what we ultimately aim to do is to use all of that information in terms of understanding test compounds binding to our protein to make design hypotheses to make new and improved ligands with better affinity, efficacy, and physicochemical. So why should we use biophysical methods to help us to do that? Well, because we can detect, identify, and characterize molecular interactions. So in detecting, we can use biophysical methods for screening, gives us a yes or no answer, does the compound bind to the protein or not? In identifying binding partners, so we can look at our protein and see whether it binds with other proteins, peptides, small molecules, fragments, and we can look at understand the specificity of those binding interactions. And then finally, we can actually characterize in more detail those interactions in terms of either the protein stability itself, the affinity of the binding interaction, the kinetics of how rapidly does our test compound associate, dissociate from the protein. Then look at the thermodynamics, so the enthalpic and entropic contributions to the binding, and then the mechanism. So understanding whether there are requirements for other ligands or potentially conformational changes that might give us the most preferential binding affinity. So I'm going to finish just with an overview of the types of biophysical tools we have available to us. The acronyms are listed here. So we have SPR, we have LMR, we have mass spec, we have ITC, titration calorimetry, BLI, DSF, and feeder. And all of these things give us different angles in terms of measuring some of those parameters that help us to answer those questions. And so you can see the sort of ways that these techniques are applied in terms of the parameters they measure. So SPR in terms of affinity, kinetics, yes, no binding, for example. So these are the sorts of tools that Chris is now going to talk about in terms of how we apply those in characterizing proteins and protein interactions at different stages of the drug discovery process. I'll hand over to Chris.
Okay, thanks, Geoff. So yeah, I'm going to go through some project examples where we've used biophysics at various points in the process. I've tried to choose diverse examples, so sampling most of the techniques that Geoff just mentioned on the previous slide, so that you can see how we use these things. The first few examples are to do with reagent QC and understanding, really. One technique we can use to understand reagents generally is mass spectrometry, but more specifically here, I'm talking about native mass spectrometry. So this is a particular flavor of mass spec where we infuse the sample into the instrument under conditions that we aim to maintain non-covalent interactions that exist either intermolecular within the one protein or intermolecular, but multiple proteins. In order to do this, we need to make sure that the solution has only got volatile additives. So typically, we definitely don't use things like tris and sodium chloride. It won't work. We use ammonium acetate to provide some ionic strength to the solution and try to keep the sample happy. Data we get from these experiments will allow us to do things like characterize oligomeric state and also see whether things bind or are bound to the protein. We've had many projects where we get co-purification of cofactors with the targets, and that's really important to understand because if you're looking for something that's competitive with a cofactor, you don't want it to be in the sample. The example I'm going to show, though, is to do with oligomeric state. We had a project where the literature suggested that the protein existed as a distribution of oligomeric states. So we wanted to try to understand if that was actually the case. So this mass spectrum shows data that was recorded in the protein in ammonium acetate. So it gives us a native mass spectrum. And hopefully you can see on the x-axis is the mass spectral charge ratio, the y-axis is just the relative intensity, and we see three clusters of sets of peaks. And these correspond to three distinct charge state distributions. With the mass spec experiment, we can deconvolute these peaks to figure out what the mass of the species was that gave rise to those peaks in the first place. And it turns out this leftmost set of peaks corresponded to the monomolecular weight, the middle one was the dimer, and the far right was a tetramer, which was consistent with what we were expecting from literature, and also a very bumpy size exclusion trace when we did the purification. This gave us confidence this reagent was fit for purpose going forwards. Another mass spec technique that we use at various points in the process is HDXMS, which you will have heard about in one of the previous lectures. But essentially, this is a technique that we can use to understand or get an insight into the dynamics of the protein and whether there's folding in the sequence. We do this by measuring the incorporation of deuterium into backbone amides if you expose the protein to a solvent that contains mostly D2O versus H2O. So if you've got secondary structure in the protein, these backbone amides will be involved in hydrogen bonding in the structure. So they're much less solvent accessible and available to be exchanged. So they will exchange more slowly. If you have a very flexible part of the protein or intrinsic disorder, you don't have that same limitation. They are very solvent exposed and they will exchange quickly. So by doing this, we can get insights into the structures of the protein. We had one project where the construct was made for it, and it has an N-terminal DVD that we expect to have structure, but the rest of the protein was predicted to be disordered apart from some reports of maybe having some helical content in the C-terminus. So we wanted to see whether we could confirm that or not. So we ran an HDXMS experiment on this protein sample. This plot shows on the y-axis it's how much deuterium was taken up by the different residues. And each of the lines in the plot is a different incubation time with the deuterium. So in a folded part of the protein where we should have different and measurable rates of incorporation, you should see differences at different time points. And that's exactly what we see in this DBD region here. That at the earlier time points, there's less uptake compared to the later. The complete opposite is true of the right-hand side, the C-terminal part of the protein, where even at the earliest time point, we see complete exchange, which suggests that there is no water in that part of the protein. So we don't see any evidence of a stable secondary structure in that C-terminus, which was important to understand for this particular reagent. And then the final example of reagent QC is the classic ITC, isothermal titration calorimetry. It's a great technique for measuring affinity in thermodynamics, and it does so by measuring heat changes when things interact. To be honest, we don't use it that often because it needs significant quantities of protein in order to do. And especially with the more challenging targets we have at the moment, it's just not available. But when you can do it, it is very powerful. So the data I'm showing is for a project where we wanted to confirm that the protein was binding competent and also confirm that a literature reported peptide was going to be a good tool for us to use to develop our assays. So you can see that on the top portion of the figure on the left, that's the actual raw data as it comes from the instrument. So each of those peaks is an injection of ligand into the protein in the cell. And as it binds, there's a heat change. And as you start to saturate the binding sites, we see this signal decreases over time. We can fit that data and determine the parameters for it. And it came out with a KD of 600 nanomolar, which was consistent with the literature. And we had an N value or stoichiometry of 1, which tells us that the protein reagent we have, assuming the peptide concentration is accurate, is completely competent for binding because we expect it to be a one-to-one interaction. So this was good data because it allowed us to use that tool peptide in confidence to help develop other assays. The next example is fragment screening, which Geoff touched on earlier. Fragment screening is a particular kind of hit finding where we screen small compounds, which we call fragments, that are generally less than 300 Daltons molecular weight. Because they're small, that means that they have relatively limited opportunities to make interactions with the targets. So we can expect them to have weak affinities. That's not always the case, and you can get some very potent fragments, but generally speaking, they are weak binders. What that means is we need sensitive methods and robust methods that are amenable to using high concentration screening in order to actually find the fragments in the first place. The two methods that are most commonly used for fragment screening are NMR and SPR. I'm going to give you an SPR example because that's my field, so why would I not talk about it? Just as a reminder, SPR is a biosensor technique where we have the target immobilized on a sensor chip and we flow the analytes across to see whether we have binding or not. In the example of the project that we did some fragment screening in, it's a pretty typical example, to be honest. So because it's a relatively large number for biophysics of compounds to be tested or fragments, we do single concentration screening. The library at this point was about 2,700 compounds. We screened at 300 micromolar as a compromise between having enough fragment there to get some occupancy to detect these weak binders, but not so much that we start seeing loads of non-specific binding. And the data from that fragment screening is shown on the slide. So in this plot, we've got each of the fragments points are shown on the x-axis, and then the responses at that single concentration are plotted on the y-axis. The data have been normalized for relative to the response we would expect to see if the particular fragment was saturating the surface. So we shouldn't, in theory, have anything that's greater than 100%. Obviously, we do, which I'll come back to in a minute. But this was a particularly good fragment screen. We didn't have that many bad actors, which I've called irregular in red. There were 94 actives in this case, which we were defined as ones that were significantly different to the median response of all the rest. So this is a nice place to be because it allows us to do follow-up of the compounds relatively straightforwardly. What does an active look like, though, in terms of its sensograms? Well, I've picked this compound here, which has a middle-ish signal on there. And this is a sensogram that gives that point there. How can I change the pointer? I can't see the mouse. So on the sensogram, you can see x-axis is time, the y-axis is the response. Just before the 0 seconds is where we have just buffer flowing across the surface. At 0 seconds, the instrument injects our fragment across the sensor chip surface, and in this case, it binds. So we see an increase in signal. It injects it for 30 seconds. We see it's a steady state, very rapidly reaches a steady state. And then the instrument switches to flow. It's just buffer flowing across again, and the fragment comes straight back off as it dissociates from the target. And this is what we would expect to see for fragments because we expect them to have weak affinities. And once you've got those actives, you need to figure out which ones are worth following up or have a measurable affinity. So you do a titration, and then you can fit that data and you can get a KD. This particular fragment had a KD of 330 micromolar. So it's a reasonable starting point to take forward and do other screens of near neighbors, for example. But what about these irregular ones? I've picked two examples and they show similar bad behaviors. I mean, the first thing to note is the response is greater than 100. That already suggests, just from these points on the plot, that there's something going on, because if you're having a one-to-one binding, it shouldn't really be much above 100%. And these are the sensorgrams for those compounds. The signals that we actually do get from these compounds are incredibly high. I should say that the point on the plot is taken at the beginning of the injection. You can't quite see because it's a small figure. But actually, the signals are really, really high. There should be maximum of 30 or 40 response units. So we call these superstoichiometric binders. It's not a good behavior. So we definitely want to just not even think about these again. But in terms of the sensorgram shape, the power of SPR is it's a real-time technique. So we see the binding as it's happening. We see the dissociation as it's happening. And for one-to-one interaction, we should see exponential association and single exponential dissociation as well. We don't see that here. If anything, it's multiphasic, horrible, sticky, awful. These are things you just don't want to follow up. And actually, they can be a problem in your screening, because if they stick and come off really slowly, they can affect the data that comes after them. So it's not a happy place to be. But the good thing is that we can see these issues. There are other assays. If you do a biochemical assay where you're just measuring an ID50, you wouldn't see this behavior. You might just get a high hill slope or something, which in itself is potentially a bad sign. But this is powerful stuff, I think. Okay, so we can do primary screening by using fragment screening, but another very important activity is orthogonal validation of hits that come from other screening methods. So that could be a biochemical HTS, where you've got a functional assay, you want to look for inhibitors, for example. It could be a cell-based screen. But the example I'm going to show is affinity screens, specifically high throughput affinity screens. More and more, we're getting targets that don't have functional activity. They're scaffolding proteins and things like that, or they're proteins we want to develop PROTACs or something or to degrade them. So the only way we can really find hits to start with is to find things that look for things that just bind the protein. So the example I'm going to show is a transcription factor, which was unprecedented in the sense that there were no compounds known or reported to bind to this particular target, and we couldn't make any ligands really that we could use to validate the proteins. So protein science made some protein, and colleagues in hit discovery did a couple of screens using these affinity screening methods. The first one I did was affinity selection mass spectrometry, where they screened 200,000 compounds. And the second one was a DSF screen, which looks at thermal unfolding and stabilization, and that was 100,000 compounds. There were lots of extra steps that went in between that point and these 43 hits that came out, cross-validation, repeats, and also when looking at the chemistry and seeing, are these compounds likely to be tractable? Do they have good properties, solubility, purity, and things like that? But from these two screens, we've got 43 hits, which we wanted to understand, are they real? Can we characterize them? Because this was an unprecedented target and we didn't have any tools, it does make developing assays challenging because you don't have a positive control to validate the assays. Fortunately for the project, we were able to make isotopically labeled protein that allowed 2D protein observed NMR experiments to be done. The power of the 2D NMR experiment is you don't necessarily need a control to be confident in the result you see. So that's exactly what we did. We put the 43 hits through a 2D NMR assay. We tested a single concentration of 200 micromolar. The figure on the slide is an example of some of the spectra that came from these experiments. So we can see in the blue spectrum is the APO protein, and the red spectrum are the peaks from the protein plus compound sample. If the compounds didn't bind, you would expect all of the peaks to overlap, but you can quite clearly see that that isn't the case. There are plenty of blue peaks that are not sitting underneath red peaks. So that, for this particular compound, is strong evidence that it is interacting with the protein. So after all 43 of those were tested, only 3 showed the behavior we expected, indicating binding. The next step, which is always the case after single concentration testing, is dose response to see if we can determine affinity for the compounds. And 2 of the 3 had measurable affinities. They were weak, but if you don't have anything else to start off with, then this is a good place to be. So as we always do when we have a screening campaign, you find your hits in the collection, and we have a huge collection of compounds. So chemists look through the collection and identify compounds that are near neighbors, chemically similar, to see whether we already have things that may have a higher affinity for the target. And a load of compounds were chosen and tested in the same way, so single concentration, then dose response follow-up. And that ultimately gave us a compound with a KD that was actually off scale for the NMR experiment, so less than 20 micromolar, that we were then able to use. This was a breakthrough in this particular project because it gave us a compound which had a good affinity that we could use as a tool compound to develop other assays. Specifically, I'm thinking here about SPR, because SPR is much higher throughput than the NMR assays, but it's not as useful if we can't validate the surface. That compound was tested in the SPR assay, and it gave us, the data from the SPR gave us a KD of 6 micromolar, which is entirely consistent with the NMR experiment. So it was really nice to see that, this compound was confirming. And this has now enabled us to test more compounds. So either look for more near neighbors, but also when we make new compounds, they can be tested much more quickly. Another aspect of this particular project was the structure. At the moment, we don't have a ligand-bound structure of the protein, which is ongoing, but biophysics can help here as well to a certain extent. The 2 methods to be used to characterize the effects of the compound on the structure were HDXMS and 2D NMR. So in the HDXMS experiment, there were peptides or residues in the N-terminus that seemed to be protected in the presence of the compound, and that's shown by these blue horizontal lines in the plot. And the 2D NMR data at this point had been assigned, so we knew which peaks corresponded to which residues in a spectrum. So then you can figure out which residues are perturbed by the presence of the compound. And the regions that were suggested to be affected by both experiments are consistent with each other. So that information is good because it can help guide the docking until we get the structure, which I'm sure we will any day. Okay, so this last section it's called mechanistic characterization, but it's kind of a couple of examples of things that are just other. The first one is a little bit dry, a bit boring, but I think it's one of the most important things that I do as part of my job, and it's to do with potency and potent compounds. This histogram shows all of the data that was recorded for a particular project where we have a biochemical assay. So we're looking at inhibition of an enzyme. So the x-axis is PIC50, and that's simply the negative log of the IC50. So PIC50 of 9 is an IC50 of 1 nanomolar, for example. But what the point I want to make from showing this plot is that we can see that we have the bunching up of the potencies. So most of the compounds that were made by the end of this project or this stage of the project have a PIC50 about 8, which is 10 nanomolar. It seems unlikely that would happen because chemists can keep improving things, but it doesn't seem to be making a difference. And the reason for that is because all solution-based assays like this have what's known as the tight binding limit, and it's essentially a limit where even if something is a better inhibitor or a better binder, you won't see that in your data because your compound is more potent than the concentration of target. So something else has to be used to help understand the compounds that are all looking like they're about 10 nanomolar IC50. Fortunately, SPR does not suffer from that same limitation because we're in a flow-based system. We don't have ligand depletion. And you can see from this histogram, the distribution of PKDs, it doesn't have this apparent wall. You'll also see that the number of compounds is significantly lower because SPR is not a high throughput technique, especially when you get to very potent slow compounds. But it enables us to characterize things that are much more potent. The important thing to consider, though, when you have two assays like this is, do they correlate? Just because you've got a more potent binder, it does not necessarily mean that it is a more potent inhibitor. To look at that, we can correlate or plot the two parameters against each other. So you can see on the x-axis is the biochemical assay, PIC50, the y-axis is the PKD. And if you concentrate on the top right-hand corner, these are points where we don't have this tight binding limit, and you can see there is a good correlation between the two assays. It's not until you get to the tight binding limit that the correlation falls apart, but you can see there is then a nice spread of affinities in the SPR assay. And actually in the project at the time, this was the only way, SPR was the only way of the chemists knowing if the changes they were making made a difference, significant difference to the potency. Ultimately, cell-based assays were used and they correlated really nicely all the way down with the SPR. So we were able to drive the chemistry off the cell-based assays. And the final example is to do with molecular glues. By molecular glues, I mean monovalent small molecules that induce or enhance the ternary complex formation between two proteins. So obviously cereblon, methylidamide, and things like CK1L3 and things like that. That's an example of a molecular glue. We can use various biophysical techniques to help understand these systems, but the one I've chosen to show is feeder flow-induced dispersion analysis, mainly because it's one of the new instruments we've got in the lab. The way that the information we get from a feeder experiment is, as long as we have a fluorescently labeled molecule as a reporter, we can measure the hydrodynamic radius of that molecule. It's a capillary-based experiment, so the instrument pushes the indicators through a capillary, and as it travels through that capillary, you get this parabolic concentration profile as it travels through there. That is detected by a fluorescence detector at a certain point along the capillary, and you get this Gaussian-shaped time signal curve. If things have a larger hydrodynamic radius, they diffuse more slowly in this radial direction, and you get much wider Gaussians. So we can use this experiment to measure hydrodynamic radius. In terms of a project and how it's actually used, we had a project where we had a peptide and an E3 ligase that bind to each other already, but we wanted to figure out, find molecular glues that would increase the affinity of that interaction. Practically, we had a fluorescein labeled peptide that we use as the indicator in this case. So in the experiment, we have a fixed concentration of the labeled peptide, and we do a titration of the E3 ligase, and we determine the hydrodynamic radius of the peptide under those conditions. And you can see that as you increase the concentration of the ligase, we see this increase in the hydrodynamic radius as the peptide is forming a complex with the E3 ligase. And we can fit the data and we get a KD of 46 nanomolar in this case. We can then do the same experiment, this time including a glue. Fortunately, for this project, there was a literature precedent glue that we could use as a control. So we do it again with the glue in the solution, and what you see is the curve is left shifted, which is, and you fit it, gives you a KD of 7 nanomolar. So we see this increased potency of the binding of the E3 ligase to the peptide in the presence of the glue. So this enables us to validate feeder for characterizing this particular system. In the project, this assay was then used to characterize some hits from a small screen that was done using a FRET assay that looked at the interaction with the peptide and the E3 ligase. So this chart, here shows an example of the data. So DMSO only, which is a negative control, has hydrodynamic radius of 1.8-ish nanometers. In the presence of the tool compound, it increases because we form the complex. But when we tested the hits from the threat screen, we saw that actually none of them showed significant increase in the hydrodynamic radius of the protein, with the exception of one compound on the end, which actually was a derivative of the tool compound. So it wasn't new chemistry, so ultimately wouldn't be useful for the project. So although this is a negative result, it's actually really important, because if we didn't have this cross-validation or orthogonal validation or devalidation of these compounds, there's the potential to waste a lot of resource on pursuing these compounds when actually they're not doing what we think they're doing. So that's the end of the examples. So hopefully we've shown how biophysics can add across various parts of the drug discovery process. I gave some examples to do with QC, screening, validation, characterization. But I guess the more important thing is that hopefully you can see that different techniques have unique strengths and weaknesses, and really the power of biophysics comes from being able to incorporate that data with data from other disciplines, I guess, which gives us confidence in the molecules that we find and how to and gives us ideas of how to progress them and understand their mechanism of action. Finally, acknowledging everyone in the UK biophysics team, many of whom contributed to slides or examples on the slides. And I've been told to highlight this book, Laboratory Guide to Enzymology, written by 3 people from Hit Discovery, one of whom is sitting right there. I'm sure it's an excellent book because Geoff is an expert in enzymology, so I would definitely recommend looking at that. And thanks for listening, everyone.
We're happy to take any questions.
Yeah.
So you didn't mention much about specificity. Is that then addressed later down in the pipeline? Because, I mean, you could use counter screens or things like that are quite common generally for.
Yeah, often it's used a bit further down because you know, we would hope that we can build in specificity with further chemistry. So it's always a question as to, you know, how early you want to look at, you know, other proteins in the cascade, and usually it comes further down because we try to take as many hits forward as we can, do some chemistry on them, then we'll build those assays in looking at specificity and see if we can make, you know, chemical changes that you know, allow us to build in the right specificity. If we rule compounds out too early, then, you know, we're going to make changes to them. So we might do it, but it might not be decision making. It might be just annotating at that stage. Want to comment any further?
Yeah, that's true. I think a lot of the assays are really resource intensive. So if you have to build assays for 3 off targets, that's a lot of work that may not be useful until much later down the cascade. So yeah,
we try not to you know, stamp collect assays because you know, we want to build the ones that are decision making, because obviously we're in a resource constrained environment. We can't do everything on every project. So, you know, making sure we do the right things that help us to make the next molecule is what we focus on.
Yeah, so it's just a basic question about like SBI. Exactly. So how do you exactly immobilize the target? And how can you do that so widely with a range of targets without like affecting like the protein itself?
There are many ways of immobilizing targets. The one that they tell you about as the usual one is just using amine coupling. So you reconcentrate your protein on the surface of the chip by lowering the pH below the PI and use EDC-NHS. We almost never use that though, because you're cross-linking the protein to the surface, so you don't really know what you're doing to it. More commonly in our team, we use things like nickel NTA chips. So we have a his tag on the protein. We'll use a bit of mild cross-linking to keep the protein immobilized. But if we can, we will generate Abby tag proteins that we can have that are bioacylated, where we know they're bioacylated. Obviously, that biotin streptavidin is basically a pseudo-covalent interaction, so the proteins stay immobilized. But still, even though you do that, that's why the tools are so important to validate the surface, because it's not necessarily the case that you mobilize and it will still be fine. Or the protein could not be very stable. You know, this is an assay that is a real-time assay. So over the course of I don't know, at least 12 hours for an assay. So the protein needs to maintain stability. So being able to assess that is kind of important.
And usually we would try to use the same tags for immobilizing proteins for SPR as we would for other methods. So we need to immobilize proteins for our DNA encoded library screening, for instance. So if we can use the same constructs, with the same tags on, we would tend to do that because, you get, cross-validation across the different techniques then with the protein that you're using. So, if you start to make different constructs, then you have to evaluate both of them. And, if there are differences, that starts to raise questions. So where we can, we'll use a similar tagging approach for different types of methods.
Great. Okay. Thanks again very much for coming across and,
Thanks for having us. Yeah,
Yeah.
happy to come and do it.
And yeah,